Alright, so I'm going to walk you through a little bit of this lab that we did last week. We had 5 samples of soil. We had our first sample, which is sterile, so it's sand, so there's nothing, no organisms, anything in it, no plant matter. Then we have 4 more cups of soil I dug up from my backyard, so there should be stuff in it. We're going to treat each of these soil samples with some sort of food and water, and we're going to test their respiration. And how we're going to do that, I filled these beakers with 10 milliliters of sodium hydroxide, which is a base, and as anything in the soil respires, it's going to change the pH of the sodium hydroxide. So sand, we're doing absolutely nothing with. The first soil sample, we're not adding any water or anything to. It's just a 0 milliliter water. Our third cup is going to be water. Now the directions don't say how much. It says just enough to get it damp, but not so much that you create mud. So I filled this just to about 50 milliliters, and we're going to see how far we can get. We just want it a little bit moist and soaking in. So out of that 50 milliliters, I used about 10. So I'm going to use 10 in my next sample because I want that to remain a constant. 15. Alright, so I got about 10 milliliters in here, and the instructions told me I needed to add about 3 grams of sugar, which I've already weighed out. And try and mix that up a little bit, but probably not a whole lot will happen. And that's going to be our sugar water, is going to be our treatment for number 4. Our 5th one is going to be just sugar, no water. So we have 4 different levels of treatment, same amount of sugar. All right. So we have our control group, and we have our experimental group, each with a different level that we're testing. Ideally, we would do multiples of these, but not in this COVID scenario. So tomorrow, I'm going to put these all in plastic bags individually, so anything that breathes in here is not going to mix with the air around. And then tomorrow, we're going to test these by adding acid to them in order to see if there was any changes in the... Alright, so day 2 of this experiment, I'm pulling out the Ziploc bags that had the soil and the sodium hydroxide in it. And I've got all 5 of my cups right there. So, assuming there's bacteria and organisms in the soil, they should have been respiring and changing the pH of these samples. So, I'm going to take a few drops of what's called phenolphthalein, and this is a pH indicator. So all it does is tell me acid versus base. See, 1 drop, 2 drops in each. That one is turning all sorts of strange. I don't know why. That's not supposed to happen. It's turning milky, which should not be happening. So I have no idea what happened there. That is very not normal. But you can see how the rest of them turned to pink, and that's what we ideally wanted. My guess is there's something in there contaminating that cup. But next we're going to take these samples, and we're going to measure how much hydrochloric acid it takes to neutralize it. So when it becomes neutral, the pink color goes away and goes back to clear. Based on how much acid we use in each cup, will tell you how acidic or basic or how much they were breathing, with the exception of probably cup number 4. Alright, so this is a basic titration. I've got 50 milliliters of acid in here, the 10 in here of our experiment. I'm going to turn just slightly to start dripping in. I do like to try and give it a few seconds to react. And it's almost there. Alright, and now my solution is back to being clear again. That means everything's matched up. I used 4.1 milliliters for my sand, my sterile soil. For number 2, I'm going to do the same thing. For soil, now we're back down to a clear, no more pink. I have now gone down to 7.2, so about a few milliliters. You can do the math. Sample number 3 was the one with the water. Pink is a bad color because the stand is red. Alright. And now we're back to a clear color there, only going down to 9.2. I have no idea how this one's going to work out because it's milky for some reason, but it's still pink, so I guess that helps. And that one has gone back to the original milky color at only 10.5. And finally, our one with just sugar in it. The bubbles are getting in the way. A little bit more. All right. And now I'm down to about 14.5 as we're down to clear. So that one took about as much as the sterile sample. So that was titration, pushing that through. They got different amounts of acid because they had different amounts of carbon dioxide already being pushed in. So the ones with the least amount of carbon, the ones with the most amount of carbon dioxide, the ones with feeding bacteria, those are going to be the ones with the smallest amount of acid in order to get it back to neutral. The sterile sample and the one with just sugar should be the ones typically with your higher amounts of acid used, because they're not, there's either no organisms like there is in the sand, or they don't have the water to move the nutrients and food around. So I hope that helped you with the experiment.